All right. Um, Hello, all. Thanks so much for joining us this morning in the US or afternoon, evening elsewhere in the world. My name is Andy Bredemeyer. I am Senior Vice President here within our medical group at Velsera. I invite you to our webinar today, Tackling Complexity in Oncology Biomarker Interpretations with CGW Plus. For those of you who are not familiar with Velsera, we are committed to helping clinical NGS laboratories succeed at high-quality clinical reporting using efficient workflows to focus the attention of your expert lab staff on the interpretation of the results with the help of our really industry-leading knowledge base. And today I'd like to introduce to you Jay Peterson, who holds degrees in cell and molecular biology and applied genomics from the University of Connecticut. He served as a clinical lab scientist and later genomic analyst at the Dartmouth-Hitchcock Clinical Genomics and Advanced Technology Laboratory in Molecular Pathology. And he's been at Velsera for, I've lost track now, but some 5 or 6 years, where he's held several roles in our clinical diagnostics product group, including key work to take popular clinical NGS assays in the market and integrate them with our CGW platform. And today he serves as the Director of Product Management for the Clinical Genomics Workspace. And he'll tell us about how we've redesigned really our Clinical Genomics Workspace, CGW Plus, to tackle key challenges in interpreting biomarkers in oncology. And before I hand it over to Jay, I just want to point out that you can ask questions during the webinar. There's a Q&A button there, should be at the bottom of your screen, or depending on what platform you're on. And please ask them there, and we will address them at the end of the webinar. Thanks for joining us. Jay, take it away. Thank you.
Thanks so much, Andy. I appreciate the introduction. And hello, everyone, and welcome to Tackling Complexity Oncology Biomarker Interpretation with CGW Plus. Today, I'm very excited to be able to talk to you more about our new CGW Plus platform and the various features and enhancements that it includes to help improve and streamline your clinical genomics workflow. To get us started, NGS testing is a powerful clinical tool, but it can also present a number of significant challenges. As the number of variants that are detected and the breadth of clinical information available continues to expand rapidly, it can be very challenging to review the variants for both quality and clinical evidence, and it's even harder to review them quickly in a streamlined manner due to the volume of both variants and cases that are being generated regularly. It can also be very challenging to find the right content for clinical reporting. And finally, it can be very hard to provide consistent reporting across the various variants as you observe them in different cases, and the cases as your laboratory is generating a high volume of cases for specific genomic indications. Fortunately, Velsera's new CGW Plus solution can help address these challenges. In designing and building CGW Plus, we've incorporated solutions to these core challenges. CGW Plus introduces adaptive learning and memory features, which are extremely powerful. And we've also consolidated the workflow elements, which allow users to review all of the therapeutic, prognostic, diagnostic, biomarker associations in a single view. It also makes it very easy to reuse and save your decisions that were made in previous cases and propagate them to future cases within the same disease context. As always, we allow you to customize the report colors and layout that you choose to help individualize that report template to your laboratory or institution. CGW Plus also has a number of powerful features to really focus your attention where it is most needed. We understand that for panels and next generation sequencing, you're generating large volumes of data, and it's important to streamline that process by focusing only on that data which requires either manual review or has updated content. For this reason, we've introduced alerts that demonstrate clearly when the KV has been updated since the last time you observed that variant. It also flags variants for review based on quality, clinical evidence, and also their novelty. And importantly, it facilitates the triage of the large number of VUS or variants of unknown significance that are detected by incorporating variant pathogenicity and function information via the complement biological classification scheme. And finally, at the core of CGW Plus is Velsera's industry-leading knowledge base, which is now even smarter in CGW Plus. It includes curated content for biomarkers like TMB and MSI. It's able to interpret co-occurring variants or combinations of variants that can have clinical meaning when present together, dynamic system-generated interpretations based on your individual or institution's preferences that utilize your choices that were made during clinical evidence review. In today's webinar, I'm going to focus on the three central components of the CGW Plus workflow, each of which contains a number of new features and enhancements to help streamline and focus your workflow and also configure and customize it to your individual SOP and laboratory preferences. We're going to step through these one by one and examine how these workflow components can really support your laboratory in the work that it is doing. Let's start by looking at the first component, which is variant quality review. You'll see a particular emphasis in this section on focusing your time and attention on those variants that require manual review based, out of the large number of variants that are initially detected using most sequencing tests. We'll also show how to automate the components of this quality review that do not need your attention, so it really streamlines this process. Let me introduce you to the new variant quality review home interface. I'd like to draw your attention to a number of key important elements that we'll walk through to orient you with this interface. At the top, you'll see a drop-down menu that shows an automated, a list of different, let's say, categories or buckets, as we'll call them, which are groups of variants that are automatically sorted based on pre-configured really rules and filters that have been set up during panel setup. In addition, you'll see that variants are organized underneath these buckets by type. In this particular example, there are small variants, fusions, copy numbers, and splice variants, but that will of course change depending on the sample that you were reviewing. I'd also like to introduce the powerful and completely redesigned variant table. This is really an exciting new component of CGW Plus that has a number of different customizable and configurable components, which will really help you mold this variant table into a form that will best support your SOP and your laboratory practices. You'll see that by customizing columns, you can decide which columns are pinned or frozen so that when you scroll, they stay visible. You can select columns to filter on the fly, meaning you can do individual filtering based on these inside an individual case. And finally, there's a new review status which allows you to mark a particular variant, whether it's been reviewed, passed, failed, or in this case, if it's an artifact. But we'll talk a little bit more about that later. Let's take a closer look at the redesigned variant table that really allows a tremendous amount of flexibility and configuration to your individual laboratory's workflow. By opening up this customized columns pane, you can select the individual columns that you'd like to pin or freeze onto the table. You can also select to remove or add different columns, and you can even reorder them, again, to customize this to the preferences of your individual laboratory. This is also available fully via self-service, so you don't need to reach out to our team to make any of these changes. You're able to do it on yourself and change it if your preferences or your laboratory SOPs are updated. I'd like to dig into these, what I mentioned are the categories of the individual buckets. In this particular example, there are 3 different buckets that have been configured. Though these can, of course, the buckets themselves and the filters around them are completely configurable and can be adapted to fit the needs of your laboratory and conform to your SOP. In this example, we have 3 different buckets, which are variants that need manual review, variants that pass quality review, and variants that failed quality review. You can see via this pie chart, it demonstrates the number of variants that automatically fall into each one of those categories. You'll see the largest fraction are variants that have failed quality review, or those with low confidence variants that do not meet your analytically validated thresholds. These are variants that don't require you to look at. However, if you'd like to, you can view these variants within this individual bucket. There's also variants that pass quality review. Again, those that meet your predetermined thresholds and will proceed automatically to the next stage in the process, clinical evidence review. And this is a smaller but still significant fraction of variants. And finally, the smallest fraction of variants, which is those that require manual review. This workflow really focuses your attention on those variants that require manual review to limit the amount of time and increase the efficiency of your workflow. These are moderate confidence variants that are slightly below your high confidence thresholds that a user may decide to rescue and bring into the next stage of this process, which is clinical evidence review. I'm also very excited to introduce the concept of graph filtering into CGW Plus. This is a dynamic and interactive graphical filtering capabilities. It lets you visualize your filter configuration and to actually see in practice how your variants filter through different conditions and the decision tree into your final filter. You can very easily and clearly see which variants are which by clicking on each individual node or component in the graph filter. You can also set up these filters ahead of time and automate them. So it gives you the power to open these up to see where your variants are being filtered in the decision tree, but also to fully automate this process. So once you configure this to your SOP and your laboratory's preferences, you don't need to edit these on a case-by-case basis. It really creates a powerful way of streamlining and essentially configuring the system to meet the individual needs of your workflow and SOP. I'd say the key themes here are in automation and transparency and customization. The transparency to see not just what's within the final filter, but to very clearly see along the way which variants matched or did not match each of the individual filters within the nodes. If you select one of the individual nodes, you can also look at the and visualize the variants that either matched or did not match this particular filter node. In this example, you'll see that in this particular node, this is SNV and indel filtering based on variant frequency. By selecting the matched, it'll open up the variant table below that demonstrates the list of variants that were matched, the 6 variants in this case. For our first demo today, we're going to look at the quality review portion of the workflow and the enhanced variant table. So to start, you can open up the drop-down menu that I highlighted previously to show the 3 different buckets that have been configured for this particular example. This is variants that need manual review, variants that automatically pass quality review, and variants that automatically failed quality review. This again stratifies the variants into manageable buckets and allows you to focus your attention, in this case, on variants that need manual review. Looking underneath this, you can see within this bucket, there are subcategories for this particular example, including small variants, fusions, copy numbers, and splice variants. You can select each of these different categories to see the variants contained and move very quickly between them to evaluate and look at these variants. Looking at the variant table and the configuration as well, you can also customize and configure the table to meet the needs and the standard operating procedures of your laboratories. For instance, this shows 3 different columns have been pinned or frozen on the table so that they don't move when you scroll through the table. You can add additional columns to this, and we can demonstrate what it looks like when you move through the variant table by scrolling to the right and to the left. These 4 columns remain frozen. This allows you to pin the key information and make it readily available as you review the case. In addition, you can add or remove columns. By removing columns, it will remove it from the column table. You can also drag and reorder the pinned columns to make the information ordered in a way that makes it easier to review, again, based on your individual laboratory or institution's practices. You can use review status to mark these as passed, failed, or mark it as an artifact. So in future cases, it'll automatically be identified as an artifact and filtered out, not requiring any future manual attention. In this case, we will mark this variant as failed as an example, and you'll see as we select this, it's updated in the user interface. Looking at this particular example, now that this variant has been marked failed, we can also jump into our graph filter and open this up on demand and investigate the individual nodes along the decision tree that feed into the final filter. In this particular node, you'll see that there are 5 variants that have matched this filter. We can select this and view these variants below in the variant table. 6 that did not match. Again, we can select this, including the failed variant that we just reviewed. And for small variants, we can select the final filter and show the 12 variants that are present. This provides an unparalleled level of transparency and configuration into the variant quality review process and how you can configure these to meet the needs of your laboratory and conform to your individual workflows. These can also be edited via self-service in your laboratory so that they can be updated and changed without the intervention of our support teams, which gives the full power of this feature really at your fingertips whenever it's needed. Now that we've reviewed the variants in this sample for quality, let's take a look at those samples within clinical evidence review. This is the second portion of the workflow, and really the goal or the key concept within this portion is to determine which variants are important to report and why they are. Laboratories often differ in the way that they can approach this process, and CGW offers the flexibility to support your individual workflow through a really intuitive and flexible UI with a host of new and enhanced features that we'll step through and demonstrate in the next part of this webinar. CGW Plus truly empowers you to implement your laboratory SOP for evidence review in a standardized and streamlined fashion. So welcome to the new home screen for clinical quality review. And you will look familiar as you've seen during the variant quality review initially. However, as we dig deeper into the powerful new features and tools that are contained within this portion of the workflow, you'll see really the sophisticated capabilities that have been developed to help aid you in your clinical evidence review process. We're going to start CER with the now familiar concept of automatically sorting variants that have passed quality review into the pre-configured categories or buckets. These automatic variant buckets for review are highlighted in this particular image. Below, you'll also see the categories. However, these are different for clinical evidence review. For these variants that need manual review, you'll see there are clinically actionable variants, VUS, or variants of unknown significance, or uncertain significance, I should say. The ability to, again, customize and configure the columns in this variant table to fit your needs, display the graph filter or hide, depending on your preference, or as you would like to work on it. Once you get into production cases, you can hide it. And then also, of course, review status that allow you to mark these as something you'd like to, that has been reviewed or still requiring review, or something that in this case is unevaluated. Now I'm going to step through a second demo that's going to highlight the clinical evidence review process. Again, in this case, you'll see for this example, 3 buckets have been created: variants that do not pass clinical evidence review, those that are automatically reviewed, and those variants that need manual review. We're going to start by taking a quick look at the variants that do not pass clinical evidence review filters. If you open up this particular bucket, you'll see that in all variants, there's quite a large number of them, and they're largely tier 4 variants that are likely benign or benign. These have been automatically sorted and do not require any further attention. And that's why they've been sorted into this bucket, again, to help you focus your attention on those variants that require it. We can also take a look at the automatically reviewed variants. In this case, there's 22 known VUS variants. These have been marked to include in the report based on the fact they've been previously observed in other cases within your institution and have already been reviewed. And it gives you the confidence to know that you would choose to include these VUS variants within your report. In this particular example, there are no clinically actionable variants that are already reviewed. And so we can move on at this point to our last, final, and most important bucket, those variants that need manual review. So we'll select this bucket and take a look at the categories underneath, starting with clinically actionable variants. Looking at these variants, you'll see that these are likely clinically actionable variants that have not been observed before in the context of this disease at your laboratory or institution. You'll also notice this column that lists update, novel, and other alerts. We'll dig deeper into this later in the webinar to provide more information about how these can be used to enhance your workflow. But right now, what we can do is show some of the features and functionality within this variant table, such as the AMP ASCO-CAP classification, biological classification, and the ability to edit and update these statuses live. For instance, if you would like to select this variant and down classify, you're able to do so, and it will automatically update this within the interface. You can select to add it to the report, which will then bring it to variants included in the report tab. And you can adjust the review status to reviewed or required additional review. This is particularly helpful for laboratories that have a number of individuals in different roles working on the same cases. Now let's take a look at the VUSs, or the variants of uncertain significance, which is always a challenge for many different laboratories. In this case, we have some filters highlighted at its top. For instance, we can look at the biological classification or compromised biological classification and say, let's look at all of the ones that are marked as likely pathogenic, as these might be of particular interest because there's potentially some noted biological impact. These have been classified automatically as tier 3s via AMP, ASCO, and CAP, but have been marked as likely pathogenic via the compromised biological classification. Now, if we'd like to via this interface, we can open up for a particular variant and dig deeper into this via the variant information page. Looking at biological relevance for this variant, we'll notice that it's marked as likely pathogenic, and based on the classification, because it matches the loss of function variant in a tumor suppressor gene. By selecting this, we can view the rule details in the right pane and see the information that supports this decision. And of course, we can also see the system-generated classification reasoning, which is a concise summary of why this variant has been classified as likely pathogenic, because it is a clear loss of function mutation in a tumor suppressor gene. We can proceed and use this process to triage variants of unknown significance and help decide if we would like to incorporate these or not into the report. If we would like to, we can go back and potentially upclassify or downclassify as well via AMP, ASCO, CAP classification. We can add or remove them from the report and also mark them as reviewed. Now, let's move on to the next tab now that we've looked at clinical evidence review tab, and we can look at the variants that have been included in the report. So in this particular tab, the variants included in the report, we're going to show a really exciting new addition that's to help enhance and really increase the efficiency of your workflow. And these are variant level alerts. You'll see on the right side of this or on this screen that there's a number of different variants that have been selected to include in the report, their classification, and then on the right side, a series of different variant alert statuses. You'll see 3 different ones: update, novel, and familiar. Update and novel are of particular interest because these indicate that either new or changed evidence is available since your previous review in this disease context. Novel, meaning that this variant has previously not been reviewed in this disease context, and so really all of the assertions will be novel in this context. Or familiar. This marks variants that there haven't been any updates in evidence since your last review in this disease context, and it's not a variant that you need to spend a lot of time on for clinical review. This is extremely powerful as an adaptive feature because the more cases and the more variants that you review, the more that will appear as familiar. It also just allows you to very clearly see novel variants and also when information has been updated so that you know where to spend your time and where not to spend your time, which becomes increasingly important and really increases, again, that efficiency and streamlines your workflow. So now that we understand that what these variant level alerts are, let's take a look at this first variant that's marked as update. This is a BRAF V600E mutation. By opening up the variant details page on this, we'll see the 4 different tabs. In this case, clinical relevance, biological relevance, or the compromised biological classification scheme, clinical trials, and interpretation. We'll take a look at these other tabs later, but for now, let's focus on the clinical relevance tab, which really shows the assertions that support this variant. In this tab, you'll notice that in the first couple columns, there's for each therapeutic, prognostic, or diagnostic assertion, the relevant tumor context and supporting sources of evidence are available at a glance. They're listed in the first, or I should say the second and third columns. In addition, you can select each of the assertions as has been done on the first one, and on the right side, you'll see a pane of information open up that provides additional details about that assertion. You can also very clearly and easily toggle off clinical assertions via the green toggle button, and you can decide to include them in your report or to save this decision for your next case using the blue bookmark. This can be really helpful because you may want to include this assertion for a specific case for a specific patient. However, you may or may not want to include this assertion moving forward in future cases when this variant is identified in this disease context. I'd now also like to draw your attention to this first column, which shows a number of different icons or alert icons. That's another type of alert that's here to enhance and support your workflow. These alerts are called assertion level alerts. And what these do is essentially they show when information has changed since the last time you viewed this variant. In this example, we see a couple of different types of assertion alerts. The first, this blue star, it indicates that this is a completely new assertion since the last time this variant was observed in this disease context. In this example, this is a new NCCN assertion, and the information can be displayed on the screen to show more information about it. You can then decide if you'd like to include or not include this in your report, as demonstrated in the previous screen. Furthermore, there's the yellow exclamation point, and this assertion shows that information has changed since the last time. So while it might not be a completely novel assertion, there is some new information that likely requires your review, and at this point, even if it was included previously, you may decide you no longer want to include it, or now you want to include it in all reports going forward, or for this particular report. Using these different variant and assertion level alerts, I'm going to walk you through an example case that can show you how these can really help facilitate your workflow. Now, this is an artificial case and has two different variants that are contained within it, as well as a third concurrent or combination. This has a copy number gain in HER2, a PIK3CA mutation, as well as a co-occurring or the interpretation of these two variants together. So the copy number amplification and PIK3CA mutation. We can see the different variant level alerts on the right side. One is update, one is familiar, and one is novel. We can focus our attention on the updated and novel while having confidence that we have reviewed all the information and nothing has changed since the last time we observed this mutation in this disease context. So starting with our updated information, or I should say, you can also mark that variant as reviewed or the status of that as you proceed. So let's look at our first variant that's for update. You'll see on the first two assertions that are listed for the FDA, one is the blue star, indicating that this is a new assertion since the last time this variant was observed in this disease context. You can select this assertion, view the details on the side, and it will even describe at the top that this is a new assertion since the last time it was seen in this disease context. For the second assertion listed, again from the FDA, this is one that has been updated since the last time it was observed. And it provides information at the top that shows what has changed. In this context, the support of the source NCCN is now removed for this assertion. Again, these are extremely powerful and helpful tools in understanding what has changed and what is new since the last time this variant was observed, so that you don't have to spend your time reviewing the other assertions that are listed. Now let's take a look at an example. Oh, and of course, mark it as reviewed as you're finished reviewing this variant. Now let's take a look at the last, which is marked as novel. So this is a copy gain mutation in HER2, and as this is novel, you'll see the blue star indicating that essentially all the assertions here are novel. You can go through them one by one, decide again whether to include them or not in this report, and whether to propagate those decisions forward. You can open them to look at the individual assertion details and really drill into this clinical evidence to give you the confidence you need in this variant. Now that we've covered that individual clinical relevance tab, let's take a look at the biological relevance tab. This is how we've incorporated the ComproMed biological classification scheme into CGW Plus. This serves as a secondary classification scheme to the CAP-AMP-ASCO and is based on biological activity rather than clinical actionability. Biologically and functionally meaningful variants often have a clinical meaning as well. The functionality can help predict pathogenicity downstream and is particularly useful in the context of evaluating variants of unknown significance or uncertain significance. Even when there isn't explicit clinical relevance known, such as therapeutic or diagnostic, this info can help you decide whether a tier 3 variant should be included into the report or not, and is a powerful tool to help in this process. In this particular example, we're going to see an AXL variant, which is marked as a VUS at the top via the AMP-CAP-ASCO classification scheme. In addition, looking at the rules, we see it matches the non-loss of function variant with low parameter score. By selecting this, it'll open up a side tab or a side panel that will show the evidence that supports this decision, essentially the component scores that add up to this total parameter score. In addition, for each variant, you'll also see the system-generated classification meaning, or a clear and concise description of the ComproMed biological classification for that variant. In this case, it is classified as a VUS as well because it is not a clear loss of function mutation, and the ComproMed score, parameter score, was less than 2. I'd also like to take a look at the clinical trials with a completely redesigned clinical trials tab that makes it easier to identify the most relevant clinical trials for the particular patient and case that you're reviewing. Very clearly, you'll see highlighted here that there's a list of clinical trials that have been included in the report in the upper half of this pane, and the lower half, those that have not been included. And you can see that this includes the closest trial site as well as the closest distance. The default ranking weighs this phase information as well as the distance to prioritize these clinical trials, and this prioritization scheme is also configurable based on your individual preferences. You can proceed to the ranking on the side, and based on your institution or laboratory preferences, you can also reorder these clinical trials from the default listing. For instance, make this number 2 trial now the number 1. You can also decide whether or not to include each trial in your report by toggling this green lever, and you'll see at the top half again, these are the clinical trials that are being included in this report. And on the bottom pane, there's additional clinical trials that you may have opted to not include for various reasons. And finally, the last tab within the variant details I'd like to highlight is how we've reimagined and improved the enhanced interpretation, generation, and review process. So let's take a deeper look at this. Interpretation is really a core component of the reportable variant review process. It is the smarter and it involves now system-generated interpretations that are significantly smarter. CGW will now automatically draft interpretations for you dynamically based on the clinical assertions that you have marked for your report in previous review components of the workflow. This text is then organized in the right panel you'll see into discrete sections that are ordered by priority, much like the clinical assertions. You can use this generated text as is, or you're also free to modify it if you choose in the editor. In the left pane is the editor, which is what you will sign out and then will automatically populate in the report and then automatically populate in the editor the next time this variant is observed in this disease context. Again, the system-generated interpretation is dynamically updated, and as you open an interpretation, even though you'll have your previous interpretation populated in the left pane, if the evidence has changed or there are new assertions since the last time you signed out a case, you can then copy and paste this new information from the system-generated interpretations into your interpretation editor on the left. This provides the most real-time updated information to make sure that your interpretations are constantly incorporating the latest clinical evidence. And at this point, now that we've looked at the clinical evidence review process, we'd like to take a look at the report finalization and sign-out process. We offer in CGW Plus a number of optimized reporting capabilities. CGW Plus can be configured for reporting to allow you to decide which KB sources should be included as on or off by default, the ranking of the assertions in your report, which trials to display by default based on your geographic region, what to prioritize in the report, and which and how many clinical trials should be included by default. Again, this comes back to the theme of consistency, which is really key when you're providing clinical reports for a specific test for a variety of different patients, to sets of clinicians or other essentially those that use the reports from your laboratory. Ensuring that consistency in both the way the information is presented and how it's interpreted is really key to building confidence in the test that you are providing. And now I'll take one look at the report finalization and sign-out interface. On the left-hand side, you will see the editor where you're able to edit the text within the report. And on the right side, you can dynamically preview those edits in the report itself. This really saves you time as you're able to make your edits, see the final report in one interface, move back and forth between these two different screens, and then finalize your report and move on to the sign-out process. So now that we've had a chance to see the key components of the workflow, including variant quality review and clinical evidence review, I'd really like to revisit the core design considerations that we incorporated into the build of CGW Plus and the problems that we really intended and solved via these different features and enhancements and workflow elements that we've incorporated. You'll see the customizable quality and clinical evidence review workflows the powerful features and the configuration of these that allow you to shape them to your individual laboratory's reporting preferences and standard operating procedures. You can retain and reuse your past decisions to make it faster and more consistent in your reporting practices. You can also set your preferences and customizations so that as you go through, again, you don't have to configure these for each and every report. It's already been configured in your workflow. One of the key tenets, again, as the amount of data and the number of cases continues to increase, is focusing your attention where it is most needed. Time becomes a precious commodity when you're signing out large numbers of clinical cases, and these features allow you to alert when new KB content is present or when it's a variant and its information or clinical evidence is familiar and does not require additional review, and also to improve the triage of variants of uncertain significance, which again is a continued and challenge within the industry based on the number of variants that are detected and the ever-evolving breadth of clinical information. And finally, a smarter knowledge base and better draft report. The core of the platform and the driver is still our industry-leading knowledge base, which now includes content for TMB and MSI, content and support for co-occurring variants, and as well as the ability to create system-generated interpretations, which are frequently updated to provide your interpretation with the latest, most relevant clinical information. Thank you for joining us today for the webinar, and now I'd be happy to take a couple of questions.
Jay, thank you. It was great to see CGW Plus in action. We do have a handful of questions. Please do add additional questions as you think of them in the Q&A, and we'll try to cover them as we're able. So first, a question about setting filters and that configurability. So if my lab can set and change default filters in your platform, can the lab restrict which personnel can make these changes? And then same for other functions like changing tier, et cetera.
Yes, we have a really robust, role-based permission framework that we built into the platform that can allow you to restrict certain functionalities to different users. For instance, if there's your bucket of variants that are automatically failed, you can restrict who is able to go into those and look at those and potentially rescue a variant, depending on the role in your laboratory. There's a lot of different ways you can configure that and really make the ability to configure it to the role within your laboratory and your individual SOP or workflow.
Thanks. A couple questions about content from the knowledge base. So on clinical trials, for clinical trials recommended by the system, does it take location into account so that I can see trials near to my patients?
Yes, it does take that into account in the prioritization and the ranking of these trials. The logic that's used to prioritize those trials and which are pulled in is also configurable based on your preferences. So you can set geographic location and then see trials that are most relevant to your patient population or individual patient if desired for a case. So yeah, there's a lot of configuration into not just selecting the trials that's scientifically right for your patient, but also ones that are geographically and are something that they'd be able to travel to for the course of their treatment.
Thank you, Jay. On similar vein on knowledge-based content, so you showed drugs that match from your knowledge base. Does this content match in a tumor type specific manner? And I guess maybe you could extend that to clinical trials too.
Yes, it's a great question, and the knowledge-based content really is it's matching content. It is prioritized based on the disease or tumor indication, so it is structured in a way that returns really those within your disease context KB knowledge with a preference. Although it does also, capable of showing, within the context of other diseases or other indications. So really the important thing is to show kind of the full breadth of information, allow you to configure and customize which of that information you'd like to see, and try to present it to you in a way that's prioritized and make the most valuable information presented up front. Although we have our resident KB expert, Andy, here as well. So if there's anything you'd like to add to that, please feel free to chime in.
Thanks, Jay. Yeah, I guess I would, that's all accurate. And I guess I would say that, where where there's evidence from a different indication, a different tumor type for a biomarker, if that's the only evidence available, that's used according to your AMP-ASCO-CAP classification, right? To place that alteration in, say, tier 2C, if this is evidence from a drug label or a clinical practice guideline. So yes, definitely preference and order of information presented to you is going to favor things that are relevant for the indication, and then other stuff is also discoverable if you want to use it.
Great, thanks. I had a feeling you'd be able to say that much more concisely.
Not sure that was concise, but thank you. If I want to use information from NCCN and FDA approved drugs, but not other sources, can CGW do that? I guess like European sources, if I don't want to look at those, but I want to instead use US sources.
Yes, you can configure your the sources that you're using for your assertions. As you mentioned, in the question, that's important depending on your geographic region and also just on the reporting preferences of your individual laboratory or institution. So one of the tenets we had of designing this is we wanted to include a lot of flexibility in configuration. So to be able to configure and choose sources and various other kind of elements within the platform, so that based on not even just your SOP, but just your preferences in a lab, institution, or region, you're able to select and make those choices so that for each report, you're getting that kind of consistency in the most relevant information that you're looking for.
Thanks, Jay.
Yeah, sure.
One broader question about I guess supported assays. So what assays can be run in CGW Plus?
Great question. At this point, we're supporting all the solid tumor indications, as well as that includes ctDNA as well. What we're actively working on and planning is bringing over also support for heme indications as well as germline. And so, CGW Plus will become a application in disease disease indication agnostic platform supporting all of these applications. I should say in the meantime, heme and germline are still, of course, supported on the CGW Classic platform. However, we will be bringing those applications so you can take advantage of all the new great features and enhancements that we include in CGW Plus in the context of those diseases as well. So more information to come as we lift over those applications and support them in the platform.
Thank you. There's a question about this example of a co-occurring ERBB2 amplification and the PIK3CA mutation and how we handle that clinical significance. And maybe I'll give that a stab in it. Jay,
Yeah,
you can add. So the Velsera knowledge base essentially sets conditions for when certain content ought to match, and that includes the ability to require that more than one alteration is present in the same tumor. And so in the case that Jay showed, there was evidence available for the presence of an ERBB2 amplification, I think, in that disease, in that tumor type, a PIK3CA mutation, a small variant in PIK3CA. There's also evidence that is unique to the condition when both ERBB2 amplification and PIK3CA are present in the same tumor. And so that is called out in CGW Plus as a distinct finding. It is, two variants that have been shown individually, but now they have a different meaning when present together. And so that clinical significance, that clinical evidence that is expressed by a clinical practice guideline, a drug label, even a clinical trial, from this evidence is displayed distinctly in the interface. So you can report that as a separate finding if you wish, and tier it accordingly and provide your interpretation of that co-occurring alteration or co-occurring set of alterations as a distinct finding that may yield some additional treatment options that are not available when the alterations are present separately.
Great. No, nothing to add. Thanks. Well addressed.
Okay, one other is on report appearance. So for current CGW customers, if they have a report they like, can they keep it when they upgrade to CGW Plus?
Yes, I mean that's one of the nice benefits of CGW Plus is that our productized report templates have been brought over so that you can really see a seamless transition to the report template that you're currently using. I will say for clients that have customized report templates, it'll likely take a little bit of additional time to bring over customized elements or to adopt if you choose elements of our productized reports. But that being said, the intent is certainly to continue to support your existing report template so that your customers and the users are have the same familiarity with that. So you get to take advantage of all the different features and elements, and we'll continue to iterate in adding new features and elements into those report templates. But you should have a seamless transition between the platforms.
Thank you. One last question I see is on knowledge base updates. In CGW Plus, how often is that content from those sources updated?
I think with our resident KB expert on the call, you can probably expound on this one. So please.
Sure. We add content to our knowledge base, and on a monthly basis, we fully verify and validate that content through a formal testing and release process. And then each month there is a release of the knowledge base that incorporates updates from all of those sources that Jay described, drug approval agencies like FDA and the European Medicines Agency, clinical practice guidelines that we discussed, clinical trial sources, and the published literature. So all of that is aggregated and packaged and released on a monthly basis and updated then in CGW Plus.
Great. Thanks, Andy. All right, I guess.
That is all the questions we have. Thanks for asking those. Thanks for your participation and attendance today. With that, we'll wrap up. Have 10 minutes back in your hour. Jay, thanks for a terrific presentation, and thanks all for being here.